Hello everybody, welcome to the 13th webinar of our CellTalk. Today our presenter will be Dr. Nupur Biswas, who is a senior bioinformatics scientist at Renix. She is working on bioinformatics as well as multiple projects which include designing personalized neoantigen vaccines for cancer patients and exploring genes behind chondromas and the health applications of digital healthcare variables. Dr. Nupur has a doctorate in physics and her research work was focused on confined polymers. Before joining Renix, she worked at Indian Institute of Science, Raman Research Institute, Collatree, and Indian Institute of Chemical Biology. At Renix, she has gained experience not only as a research mentor, but also a scientist contributing to various international publications. So here we have Dr. Nupur. Over to you.
Thank you, Divya, for your nice introduction, and welcome everybody in my today's presentation. Today we are going to discuss about a paper which is on somatic mutations in Alzheimer disease neurons. This paper is originally authored by Michael B. Miller, and other people are also there. Michael is the first author of the paper. The group is from the Harvard Medical School. And this paper was published this year, in the beginning of this year, in the journal Nature. And it is about Alzheimer disease. So today we are going to talk about the Alzheimer disease. And although the research is on Alzheimer disease, it has quite broad impact. That's why we are going to talk about this. It has impact on the cancer, and it is actually exploring the genetic landscape of the disease. So the methodology, whatever is followed here, is applicable to in any genetic disease in general. So coming back to the Alzheimer's, what it is? Alzheimer's disease is a brain disorder, and it is usually observed among the older adults, that is quite among the aged people. And the symptoms includes the loss of memory, thinking skill, and deterioration in cognitive function, which actually makes the lifestyle difficult. It's difficult for the people to do the regular work. And of course, it is a disease of concern because globally, according to the WHO website, that almost 35 million people are affected. Now, regarding the word Alzheimer, this word Alzheimer came from the name of a doctor. It is named after Dr. Alois Alzheimer. In 1906, that is almost more than 100 years ago, Dr. Alzheimer detected some unusual clamps, tangled bundles of fibers in the brain tissue of a patient. That patient died of mental illness with the symptoms of memory. Now, those clamps, fiber bundles, whatever Dr. Alzheimer observed, that is more than 100 years ago, are still considered the major hallmark pathologies for the Alzheimer disease. The clamp or the plaque, whatever he observed, that is the extracellular plaque deposits of the amyloid-beta peptide. Amyloid-beta is the peptide which is well researched, and there's lots of work is done on these peptides. That is the main causing agent of the disease. It is produced through the proteolytic processing of transmembrane protein that is called amyloid precursor protein. In brief, we write it as APP. And the deposition of the amyloid beta peptides, which actually oligomers and create this plaque-like thing, this deposition actually disrupts the blood supply to the neurons. And that's why the neurons fail to function properly, and symptoms like memory loss, loss of thinking power appears. And the tangles, what was observed, that is actually flame-shaped neurofibrillary tangles of the microtubule binding protein tau. Tau regulates the assembly and maintenance of the structural stability of microtubules. Microtubules are the building blocks of the cytoskeleton. But in the diseased brain, this tau becomes abnormally hyperphosphorylated, and that causes the microtubule to disassemble. These pre-disassembled tau molecules then aggregate into helical filaments and produce some tangles. Now, with this background, the present challenges with the disease is to understand and really know for better understand the specific event that cause the neuronal dysfunction and death. The landscape of the disease, which will help us to provide effective immunotherapy. And from these challenges, the objective of the current work is set to study the mutational landscape of Alzheimer's disease, time evolution in the somatic mutation, that is the effect of aging in the mutation, and achieving single cell resolution, and to explore the mutations. And of course, to study the consequences of the mutations and how the disease prognosis occurs. The methodology, whatever is followed in this paper, the major one is the single cell whole genome sequencing. Single cell whole genome sequencing was performed on multiple sites, basically 2 sites of the brain's tissue samples were collected from the prefrontal cortex (PFC) and the CA1 subfield of hippocampus region. This one is the CA1 region and this is the PFC region. Cells were now, this is the cells from the brain, so it is very difficult to collect it from a living person. So the cells were collected during the post-mortem from 8 Alzheimer patients, 18 controlled patients. Multiple cells were collected from each patient and multiple cells were also sequenced. Okay. And for single cell whole genome sequencing, for the amplification process, for the genome amplification process, they primarily use multiple displacement amplification method. This figure shows the schematic of the methodology. For the both control and Alzheimer's disease, tissue samples were collected from these 2 regions. These are the nuclear isolation was done. Then neuron marker was used to identify the neurons. Then genome amplification followed by single cell sequencing, and then bioinformatics analysis was done. And another methodology they used is the immunofluorescence microscopy. For that, they use one molecule, that is 8-oxyguanine. 8-oxyguanine is quite common. It's common DNA lesson. So it results from the guanine modified by the reactive oxidant species. It is actually basically oxidized form of the DNA, guanine site. And this is used as the biomarker for cellular oxidative status, and it actually marks the DNA damage. And immunofluorescence microscopy was done using antibody targeting 8-oxy, and NeuN is also used to label the neurons. Now, what they found is, first of all, they found how the mutations are evolving with age. Here, the graph shows the result, the primary result. The blue is from the control and red spots are from the Alzheimer's disease patient. We see over the age, the blue data is available for less than 1 year to more than 100 years for the control patients. But for the Alzheimer's patient, data is available only after 60 years age. That is because the Alzheimer is a disease observed in the elder aged people. But what we see, yes, the blue circles are for the PFC region and triangles are from the age region of the brain. For Alzheimer's patients, the values are quite high, both for the red circle and the triangles. But that means for both the region, amount of mutation accumulated are more in case of Alzheimer's diseases, and it's quite more in the hippocampus region. And they also studied about the mutation signature analysis. Mutation signature is actually characteristic combination of mutation types, right, for arising from different but specific mutagenesis process. But mutagenesis signature A, B, and C, they actually primarily identified from their earlier studies, and what they observed is signature A has increased in all samples. The first graph, signature A has increased all samples. Signature C has also increased here, but increase in signature C is more in case of the Alzheimer's disease. And the spread is also quite high in here. In this graph, it is visible quite nicely. But signature A spread is quite less. From this thing, they what conclude that signature E represents age-related mutations, which are obvious both for the norm control sample space. But signature C is from the irregular calamitous events possibly associated with the disease. And this is the immunofluorescent microscopy result. The left block is from the control sample and the right block is from the Alzheimer disease patients. The green spots are actually marks the neuron. It is coming from the neuron marker. And we see some light violet color spots here and here also. Those are from the 8-oxy. The upper panel shows only the 8-oxy positions in the white color for better viewing. They have done such representation. What we see, in case of the Alzheimer disease, more white spots are there. That means more oxidation in case of Alzheimer disease neurons. Okay, this is a quantitative presentation of the result. They measure the quantitative presence of the 8-oxy, and with age we see that for control it is increasing. Of course, it has increased, but for Alzheimer disease, the increase is more. The values are quite high. That means more oxidation in case of the Alzheimer disease neurons. Then they associated the mutational signatures with the gene expression values. So signature A mutations, how it is increasing with the expression values, and this quite positively correlated. But signature C is quite inversely correlated with the expression values of the genes. And more interestingly, what they found, they actually did not find any somatic mutation in case of the classic well-known Alzheimer disease genes, which include particularly APP, PSEN1, PSEN2, and APOE. Among them, APP is the precursor gene of A-beta. And with this thing, they tried to find what is the consequences of these mutations, these somatic variants. This mutation can lead to the neuronal dysfunction, direct impairment of transcription, and alteration in the protein stability. If it's a mutation in the protein region, it can create neoantigens, and these somatic mutations accumulate in the genome. So, if there is a high probability two deleterious mutations are accumulated in the same gene, which can produce a knockout cell, and this chance actually increases as more mutations are accumulated as the patient is becoming older. This used some mathematical model also that suggests the dysfunctional neurons could be markedly more abundant in Alzheimer disease, which can result in the compromised neuronal function. And here we see one plot, but it shows that actually the mutations are more associated with the transcript regions of the DNA of the gene rather than the untranscript region. So it means that the mutations are more related, transcription-related events are causing the mutation, causing more somatic variants. And this is, okay, this results, whatever I presented today is based on the amplification method MD. Now the question remains, is it the result of the amplification? Is that causing any artifact? To resolve this, they started with a different amplification method, that is primary template directed amplification, PTA. It is a newer method. It some, not all samples, few samples with this method, and they show that the data follows similar trend with the MDA method. So their result is independent of the amplification method. It is a generalized theory, generalized outcome. And finally, they wanted to relate their result with the disease prognosis or pathogenesis also. So they proposing that actually it is the A-beta, amyloid-beta, which is introducing or initiating cascades of events. These events include, first diagram, it shows that this is the tau protein and this is the A-beta oligomers. It actually induces the conversion of the tau proteins to the neurofibrillary tangles and the accumulation of the ROS molecule. ROS is the reactive oxidative species molecule that can be peroxide, oxide, superoxide, like this type of molecules. And this ROS actually causing the damage to the site like this red mark site. And of course, there are, and cause this ROS is the responsible for the causing the DNA damage by the oxidative stress, by creating oxidative stress. And of course, there exists repair mechanism within the cell, like nucleotide excision repair complex. But the thing is, since the mutational burden is very high, the nucleotide repair excision repair complex or repair pathways fail to repair the mutation, repair all mutation sites. So the somatic mutation is generated, and this accumulated somatic mutations finally cause to the cell death. And then finally they conclude that in the conclusion we can say from this study the neurons from Alzheimer disease postmortem disease carry more mutation. Their accumulation pattern of the somatic single nucleotide variants differs from the normal aging neurons, and no somatic mutation was observed in the risk genes like APP, PSEN1, PSEN2, APOE, and also APP copy number did not increase. And it is A-beta which is inducing the cascades of events that lead to somatic mutations and further leading to the cell death. The impact of the study is first of all, it is clearly showing the relation between the aging and the mutation burden. It provides genetic mutations at the single cell level that is important definitely. And the role of the previous well-known genes are also explored. And finally, it proposes how A-beta is taking an important role in inducing the somatic mutations and finally to the neuron death. With this, I will stop here. I thank everybody for your attention. And if you want to reach me, you can write me at nupur@lenix.org.
Thank you, Nupur, for this very informative and educational talk. I hope everybody has a lot of take-home messages from this. Now we'll move on to our question and answer session, which is going to be very interesting. And I have few questions. Firstly, from Dr. Shaheen Shah. She wants to know what are reactive oxygen species and what are signature mutations.
Well, first I will talk about the reactive oxygen species. They are actually highly reactive chemicals. They are formed by different mechanisms, but basically they formed from oxygen. So they conclude some examples are the peroxide molecules, superoxide molecules. These are the super oxidative. And what they do actually, they oxidize. In this case, what I mentioned, they actually oxidize the guanine site and make the DNA damaged DNA. And your second question is about mutation signature. Okay, mutational signatures.
What is signature mutation?
Signature mutation. Signature mutation basically a combination of mutations. Mutation, we know that adenine A to T, A transformed to T, C transformed to T. Like these are we consider the mutation, nucleotide mutations. Signature mutations are a combination of mutations and arising from specific mutagenesis. The mutagenesis can be of various reasons. Like in this case, the group of signature A type mutations is associated with the normal aging thing, but the researchers are telling the signature C, another group of mutations that is associated with the Alzheimer's. I hope I can answer you.
Yeah, I hope your doubt is cleared, Shaheen. The next question is from Yogita. She asks, yes, what are clock-like mutations in human somatic cells?
What are, sorry?
What are clock-like mutations in human somatic cells?
Yes. Clock-like mutations are also associated with the mutational signatures here. That is not explored here in that way, but what this paper is talking about the mutational signatures. This signature A, B, C, what they are showing is actually they characterized in their earlier studies where basically signature, I can tell you that signature A is specifically in case of this disease scenario, signature has A has more mutation from C to T and T to C, whereas signature B has mutation from C to T only, major only. And signature C actually had all types of mutation in almost equal proportion. That's the thing.
Yeah,
and signature mutation signatures also you can see at the cosmic side of the cosmic side. They also provide the signature mutation for cancer.
Thanks, Nupur. I have one more question from Yogita. Can you elaborate more on the multiple displacement amplification method?
Well, this is an experimental technique. Okay, I can tell whatever I know. So it is what we get when the tissue DNA sample is collected from the tissue, the small amount of sample, that amplification is needed for the sequencing experiment. There are multiple amplification methods. Is there? MD is one of them, which is used for genome or exome sequencing. See, it is compared to the conventional PCR-based method. It does not employ sequence-specific primers. Rather, it actually amplifies all DNA. Yeah, that thing much I can tell about this.
The next question is from Dr. Vijay. He asked, were there any frequent genes among multiple cells and multiple samples, or any frequent pathways were affected?
Well, pathways have not been reported. Pathway analysis is not done here, but gene level information also they have not shown. What they have mainly focused actually how the mutational burden is changing with the age of the patient. That's the thing you have written. And they studied only the well-known Alzheimer disease genes like APP, PSEN1, that type of genes only they are reporting, not the general other oncogenes or tumor suppression genes. No comments on that thing, at least in this manuscript.
So, not about oncogenes and tumor suppressor genes, but the thing is, if they have analyzed so many samples and there are so many cells, and we see that there are up to 6,000 mutations in each sample. So there might be possibility that there might be common genes within a single individual cells. That will be the first part, and then different individual cells. So it might show some pattern that there might be some gene, specific gene either, or some pathway that is affected, that is implicated in the development of these signature changes in this brain of Alzheimer disease patients. So in that respect, they have not discussed anything with respect to that?
No, that is not the focus of this paper. I understand your question, and yes, of course, that will help to identify the biomarkers.
Yes.
I understand the question. Yes, of course, that is important, but no, this paper is mostly focused on the single cell data. At least that means they are most focused on the resolution. They are studying the single cell sequencing.
At least do they discuss the commonality of the mutations in different cells? Are they same or different?
Cell-specific discussion is not there. Yeah.
Okay.
I have a question from Dr. Bharti.
Yes.
What is homologous recombination deficiency in signature mutations?
What is? Sorry.
What is homologous recombination deficiency in signature mutations?
Homologous recombination deficiency, signature mutations are actually quite defined. Okay. The set of mutations, actually it is the same mutation A to T or C to T or T to C like this, but in some particular process, like normal aging is a process. In that process, some set of mutations are getting expressed. Some set of mutations are happening more. But for other some of mutagenesis, like disease-induced mutagenesis, some other type of mutations are happening more, and that's why they are separated.
Okay. Hope this answers your question, Dr. Bharti. Are there any more questions from the audience?
Yes.
Yeah. Oh, thank you, Dr. Nupur. Can we move on to the last slide?
Yes, sure. Yeah.
Our next presenter will be Mr. Abhijit Chauhan, and the topic of his presentation will be caste, religion, and mental health in India, which is going to be on 21st of September this month. So thanks everybody for being present for this presentation.